All right, section 1.4 is about experimental design. And another way of saying experimental design is design of experiments, or DOE for short. So the purpose of an experiment is to investigate the relationship between variables. And so to do this, you conduct research and you follow these basic stages. First, you design an experiment. Then you execute the experiment, you collect data, and you analyze the data. Experimental design, which is the first part, is the most important step of the research process. An error made in experimental design will likely invalidate your data analysis at the very end. So if you mess up this first step, you're going to have wasted a lot of work and effort, and it's going to invalidate anything that you calculate or any correlations you come to later. So here's an example. So let's say you're exploring the effects of an experimental treatment. Okay, and I'm using this flask symbol to kind of show a treatment. So you're testing the effect of an experimental treatment on plant development and yield. So yield would be how many ears of corn grow on a single plant, how many apples grow on a single apple tree, right? If only 5 apples grew in an apple tree, that would be low yield. If you had 50 apples growing on a tree, that might be high yield, okay? So consider two sets of experiments. So experimental design 1 versus experimental design 2. So in both experimental designs, the experimental treatment is applied at the time of sowing the seeds. So I've used this symbol, which looks like a bag of seeds. Okay. So let's look into a little more detail about what experimental design 1 is. So I'm using as an example an experimental design that's common among agronomists. So agronomists at arable research centers and farm demonstration sites will take measurements of seeding conditions and the applied treatments, and that's recorded. So here we've got, the seed, we've got the treatment, okay, being applied at the very beginning, and that information is recorded. So the plants are then allowed to grow until harvest time. So we take the seeds with the treatment, we plant them, we allow them to grow, and then they're harvested. And then at harvest time, plant measurements are made and recorded. So at harvest time, you record the data about the harvest, and you use tools and instruments to maybe make those measurements that you want to record at harvest time. But if the treatment resulted in a significant increase in the yield, the researcher will be unable to fully explain the field results. Without additional measurements taken during the growing season, let's see here, put a parenthesis there. Let's see here. A detailed analysis of morphological and physiological factors affected by the treatment cannot be performed. So additionally, if a natural catastrophe occurred before the harvest, then nothing can be learned. So you'll notice with this particular experimental design, you're only recording information at the beginning of an experiment and at the end of the experiment. So before the experiment ended, you could have had some kind of natural catastrophe that took place. You could have had, freezing temperatures or temperatures that were really hot, or maybe there was a fire, right? Or maybe there was some kind of biological contaminant. Maybe there was a fungus that spread throughout the plants and damaged them. Or flood or lightning storm, a hurricane, something came through and kind of wiped everything out. So if a natural catastrophe happened somewhere near the end, then you would have learned nothing because you didn't record any information during the planting process, during when it germinated, when it matured, and then finally when it yielded either, some vegetables, some fruit, some seed that you wanted to collect. You didn't collect any information during the experiment, only at the beginning and only at the end. Um, so by collecting information at the beginning and the end, you're only kind of connecting a treatment with an outcome of that treatment only. Okay. You won't really learn how this treatment maybe affected different aspects of the growth. Okay. Um, because you didn't record anything about the growth in order to tie it back to the treatment. All right. And a demonstrated weakness of something like this is a hydroponic study to measure the effect of varying concentrations of sulfur has on the growth of wheat was ruined when field mice invaded the glasshouse and ate the wheat 2 days before the planned harvest date. So just before they were going to harvest, right here, little mice came in just before the harvest and ate all of the crops. So because the mice came in and ate all the crops, you couldn't learn anything about the end of the experiment. So rodent control should have been part of the experimental design. So some planning should have gone in at the very beginning to prevent that from happening. If measurements of sulfur content inside the stems, inside the leaves, had been collected during the growth period, then some correlations could have been made on sulfur concentration and wheat development. So what this is saying is, had there been some measurements of, the sulfur content, in a young plant, in a mature plant, maybe how many days it took, from planting to when it actually sprouted to when it germinated. Um, if any of this information had been collected before that catastrophe took place, you would have learned something. You could have tied that to your treatment, right? Could have learned something there, but nothing was actually collected during that time. So experimental design 2 includes recording of plant measurements throughout the growth process in addition to harvesting. So, in addition to recording the initial conditions of the experiment, you record information about the planting. You record information about the germination and the early growth. You record information about the maturing of the plant. You record information about the budding and the growing of the fruit, the vegetable, or the seed that you're going to harvest. And then, you harvest, at the very end, and you make those measurements at the end as well. So had there been, some kind of natural catastrophe that happened here at this part of the process, at least you would have information on the early parts. Okay, you would have collected this early part information. And when you go to repeat this experiment, because there was a natural catastrophe, you could follow all of the same conditions that you had started at your first experiment, and then tracked the progress to see if your second experiment that you did, if the measurements in your second experiment at least match that of your first, and then keep going until the end. Okay, that would be on your second experiment. So, doing something like this, would allow you to, see, I'm trying to scroll. It's not letting me scroll. Scroll down, please. Thank you. So now when differences in the harvested plants are observed, measurements recorded during the growth process will explain the yield results. In addition, if a natural catastrophe does occur, having some measurements or records taken during the growth process can be used to still learn something important. So when it comes to a design of experiments, you need to think about what information needs to be measured, what needs to be recorded. You need to think about what resources are needed to collect the information required. How many people do you need? How many computers do you need? Microscopes, microscope slides, everything. And then what mathematical analyses, what statistics do you need? What models do you need? Hypothesis tests and so on. What things do you need to apply in order to find the correct correlations between the variables? Okay. So a lot of thought has to go into conducting an experiment before you actually conduct it. All right, to try and prevent loss of time, energy, and resources and money. In a more relatable scenario for students, consider exploring the correlation between wearing perfume or cologne and the number of coffee date invitations that a person gets. So here we've got this variable with the amount of perfume or cologne that's worn. Okay, it could be even zero perfume or cologne, versus this other random variable, which is the number of coffee dates. Maybe this is the number of coffee dates per month, right? So the question is the amount of perfume or cologne applied and the resulting number of coffee date invitations the only information to be measured? What about the type of perfume or cologne? Is it a floral perfume or cologne? Is it woodsy? Is it soapy? Does it smell clean? Does it smell musky? What about, the gender identification of the wearer? What about the clothing of the wearer? What about the environment the wearer is in? Was this, person in a coffee house, a movie theater, bookstore, Disneyland, beach, the gym? What about the type of perfume or cologne that's worn by the inviter? What about the clothing of the inviter? What about the environment? These are all factors to consider and to control for. You really have to put a lot of thought into it in order for any correlation you find to be meaningful. What information do you need to know to have confidence? And if you have this correlation, that this is actually true and meaningful, versus if you have this correlation, right? This kind of correlation here is telling you that if you wear just a small amount of perfume or cologne, you get a lower number of coffee dates per month. If you wear a lot of perfume and cologne, that'll result in a higher number of coffee dates, right? Whereas if you had a negative correlation, this actually means less is more, right? If you wear little to no perfume or cologne, that's going to correlate to having a high number of coffee dates. Whereas if you just dump a bucket of perfume and cologne on you, okay, that results in having a lower number of coffee dates. Okay, but in order to be able to have some trust in this relationship and the data that you've collected, you really have to think about a lot of things ahead of time. All right, in order to control for that. Okay, so this both the previous example and this one, are talking about the design of experiments and all of the thought that has to go into it. Let me know if you have any questions.